This pie chart is an interesting chart to help us understand why and how the body can send more blood to muscles when you're exercising. It has to do with a blood reservoir. So you'll notice that the large majority of your blood, 64%, is found in your systemic veins and venules when you're at rest. So this chart is based on rest. When you're at rest, 64% of your blood is in your systemic veins and venules. Only 9% is in your pulmonary circuit. 7% is found in your heart chambers being pumped. 13 in your systemic arteries and arterioles, and 7% in your systemic capillaries. If you were to begin exercising, we would have to be able to get more blood to your skeletal muscles. So what happens is your systemic veins and venules reduce the amount of blood inside them by vasoconstricting, whereas your systemic arteries and arterioles are going to dilate to bring in more blood to your skeletal muscles. So this is called a blood reservoir. It's a way to shift more blood into your arteries when necessary. The two really useful situations would be exercise, so you can get more blood flowing to your arteries and your muscles, and hemorrhage. If someone is losing blood because they're bleeding, we can shift blood from our veins into our arteries to keep oxygenating organs. So this is an interesting pie chart that helps us understand how we can shift blood to different parts of the body. This is a summary table. You can look this over on your own. It's in your book. But it discusses some of the distinguishing features between different types of blood vessels. But I'm going to leave this table to you. You can read through it. Some of the things you should make note of, because we talked about the differences between many of these blood vessels based on what kind of blood might be in them. This has to do with the systemic side. So you'll notice all the arteries and arterioles are oxygenated. The capillaries are doing their exchange, and all the venules and veins are deoxygenated, bringing that blood back to the heart, specifically the right atrium. But you can look over some of these other details. The big topic of this video is going to be capillary exchange. Capillary exchange is how do we get molecules and fluid exchanged at capillaries. So the exchange is occurring between blood and tissue cells. So we need certain things to go from the bloodstream into your cells. We need things from your cells to go into your bloodstream. So that is the exchange that occurs at capillaries. And many of these processes we've heard of. We've heard of diffusion. We should already know what diffusion means. Diffusion means things move from high to low, or the movement of substances down their concentration gradient. So if there's a molecule that's at a high level in the bloodstream but at a low level in tissue, it's going to diffuse out of the blood into that tissue. If the tissue is making a substance, maybe it's carbon dioxide or waste, it's going to be in a high concentration in the tissue, and it's going to diffuse into the bloodstream. So this is, the most important method of capillary exchange. It's a way that it doesn't require any energy to do this. Things are going to just diffuse if they can travel through the capillary wall. And the molecules that can do this are oxygen and carbon dioxide, glucose, amino acids, some hormones. They're going to move by diffusion. The next method we've also heard of, endocytosis and exocytosis. But together, it's called transcytosis, because you can move material into a cell, endocytosis, then out of a cell, exocytosis. So in essence, you could use the combination to move material through a cell. Remember, a capillary is just a cell wall. It's a cell that's forming the wall of the capillary. So you can move things right through using vesicles. An example is insulin. Insulin is moved out of the blood into cells or into tissue through the wall of a capillary by transcytosis. Okay, the third method is going to be the trickiest. It's called bulk flow. It's a process that moves large amounts of molecules in fluid. So this is how a lot of your ions and other molecules can be moved all together within fluid. So it's the movement of fluid, and it just so happens that in that fluid there can be a lot of solutes, a lot of molecules. Bulk flow is a two-direction process. When fluid moves out of a capillary, so from blood to tissue, it's called filtration. If the fluid is moving from tissue into blood, it's called reabsorption. Filtration versus reabsorption. This bulk flow is driven by pressures, and there's a number of different pressures at work at a capillary. We're going to learn the two most important pressures. The first one is called BHP. BHP stands for blood hydrostatic pressure. Blood hydrostatic pressure is blood pressure. But we're looking at a capillary. It's not the 120/80 that you often think of. That's at the aorta. That's at arteries. By the time we get to a capillary, blood pressure is quite a bit lower. In fact, in this capillary shown here on the left, the blood pressure at the front end of a capillary is about 35 millimeters mercury. When it gets to the end of a capillary, we call the venous end, it's 26 millimeters mercury. So that is the pressure generated by your heart. It's at a capillary, so it's not the 120/80, it's not oscillating, it's not going up and down anymore. But it is continuing to drop throughout the capillary. But that blood pressure, blood hydrostatic pressure, is a major filtration force. So it's forcing fluid out of the capillary. The major reabsorption pressure is called BCOP, which stands for blood colloid osmotic pressure. It's an osmotic pressure, which means it's pulling or it's moving water towards a high amount of solutes. And the solutes that are very important for this are the proteins in your blood. So plasma proteins are the reason why fluid will be forced back into a capillary. This is an osmotic pressure. You'll notice that the BCOP number does not change. It's 26 here, it's 26 there, it's 26 throughout this capillary. So that pressure doesn't change. But the blood hydrostatic pressure drops. These pressures allow us to learn what's called the net filtration pressure. Net filtration pressure is if you add up all the pressures at work, what's the result? So there's a little bit of math down here, but this math is just taking all these different pressures, blood hydrostatic pressure 35. There's another small one called IFOP, that's 1. So you add those two together because they're both pushing out. You subtract any pressure that's a filtration pressure because it's working in the opposite direction. So notice 35 plus 1 minus 26 equals 10. That 10 is the net filtration pressure at the arterial end of a capillary. If we compare that to the net filtration pressure at the venous end, it's much different. It's a -9. The negative means that you're now pulling fluid into a capillary as opposed to pushing fluid out. So notice these arrows. Net filtration pressure at the arterial end is a filtration. It's pushing out. Net filtration pressure at the venous end is a reabsorption pressure. It's pulling in. So when fluid comes through this capillary, we force fluid out. It delivers whatever in that fluid, like ions and other substances. Then that fluid gets pulled back in. It's different fluid, though. It's going to have waste in it. So this is a very important method to help do this capillary exchange. The problem is it's not a perfect system. It's not balanced because the reabsorption pressure is never as much as the filtration pressure. It's a -9 as opposed to a 10. If this was a -10, it'd be perfectly balanced. That would mean you're going to force out at the same pressure as you force back in. But because of this imbalance, we need lymphatic capillaries. That's this green guy here. This lymphatic capillary is collecting the little bit of excess fluid that can't be reabsorbed back into the capillary. So your lymphatic capillaries help to drain this excess fluid that will accumulate because of this imbalance we see at a capillary. The imbalance is called Starling's law of capillaries. But it's very important to have lymphatics to collect that fluid. It's also very important to have protein in your blood. The protein helps the reabsorption process as well. So this might not sound like you're going to ever need to know it, but it actually has a very, very important clinical application in that individuals develop edema for a number of different reasons. One reason is the capillary can become too permeable, and you filter out too much fluid and it can't be collected. Another cause could be high blood pressure. If someone develops high blood pressure, they force too much fluid out, they can't collect it all. Another reason is a drop in their plasma proteins. Plasma proteins are very important for this BCOP process. So if you lose plasma proteins, you can develop edema from that. One cause is liver problems, because your liver makes a lot of your plasma proteins. Finally, if there's any kind of disruption to your lymphatics, individuals can develop edema. If we were to remove this lymphatic vessel here, this tissue would have too much fluid accumulate. So surgical procedures that disrupt lymphatics, removal of lymph nodes can disrupt lymphatics, and that can all play a role in the development of edema. So very important clinical application when learning capillary exchange.